Good evening, everyone. Welcome to this new session on classification of genetic variants. And this session will be done by Dr. Bharti. And as you all know that we have been doing these online sessions in which people, or I should say, we have gathered professionals from all over the world to deliver free lectures on the topic of their expertise. This program is started under the vision of BDG Lifesciences, that is Envision with BDG. And today is the 12th session, or I think it is the 13th session on under this initiative which we have started in the month of October. So I can see there are many participants who are who have been attending these programs regularly. So I need not to tell you about the program which we are doing from the beginning. However, there are a few participants which I can see they are new. So for them, I would like to tell about this program. So first of all, I would like to, okay, someone is saying they can't hear my voice. So someone's microphone is on. Can you please mute yourself, everyone? Rajat Sahu, yeah, your microphone was on, and then I don't know, because I could see, someone's microphone is on. So okay. Okay, so I can see there are a few participants who have been who are new, and we are definitely going to start off with a small presentation of the company, like who we are and what we do, and then I'll introduce our speaker for today, and then we can go ahead and start of the session. So as far as I'm seeing in the chat box, people are saying that they can't, we cannot hear, you are not audible. Okay. So is everything fine now? Because if once you tell me if you can hear me properly, then I'll proceed. So if you cannot hear on Zoom, and if you are hearing properly on YouTube, then I think that the problem is not from my side, or the problem might be from your side. So Suma, she has already sent that it's clear for me. And yes, everything is fine. So yes, thank you very much. Thank you, participants. So quickly, I'll give you the presentation about the company, who we are, what we do, and then I will proceed with the next step. However, yes, everything seems fine. So let's start off who we are and what we do. So BDG Life Sciences is a guide who is helping young researchers and students and faculties and company professionals, and we are guiding them in a very strong way, and in a way where we where they can take our help and climb the ladder of success in their career. And especially this help is really good for students and research scholars, because the most important thing is for them what we are doing, this this is step of guiding, and what exactly it is. We are showing you the right path, and solving your queries, giving you correct training, paving your path for a successful career, and giving you launch pad to excel in the field of science. So this is what we are, who we are, what we are doing since the last 10 years. Because in 2009 we started as a group, And as you can see over here, that when we started in the form of a group, we made a group where the motive of creating the group and then transforming into an organization is simply that we want to help, and help in such a way so that you can excel in your career, so that you can achieve new heights in your career, so that you can achieve those things. Like for example, most of the people got stuck into their final year, and they do not know that what they will be doing after their bachelor's degree and after their master's degree, so to get a better job opportunity. So we are helping them as a guide, and the all these services are for free as of now, because that is the social, that is the part of the social services of BDG Life Sciences. So this is what we have been doing since 2009. Now in 2009 we created a group on Google Groups. Now back then it was Google Groups. Now the technology has changed, so we have transformed our, and registered ourselves on platforms like Facebook, and then these messengers have come like WhatsApp and Telegram and what I am. So a lot of people they ask for the they ask for our help, and a dedicated team works 24 hours to help the query. So what exactly, how we exactly started? So we started a group for students, researchers, and company professionals, especially related to the field of biotechnology, bioinformatics, and pharma. And what exactly this group had? This group has free consultation, project reports, on these PDF files of the books, and the most important thing is the job offers, that is in India and abroad, then scholarship opportunities, the PhD opportunities, and then the latest science and technology news. So this is what we are doing. Basically, we started to we wanted to fill the gap between in academia and industry, and this is what we have achieved by connecting people, so that people can avail the opportunities which are offered, and basically at the right time, because there is a huge gap between the opportunity that is shared and the opportunity that is being grabbed. So what we have done is that we have tried to fill that gap by passing on the information. So this is what was the motive of creating the group, to help the people. So as of now, we have more than 1 million subscribers, and these subscribers are not only students, but lecturers, company professionals, scientists, and professors from all over the world. So this is what we have achieved as of now, and we have our own database, and this is how we are helping people to excel in their career. So these are some of our subscriber platforms. You can see like Instagram subscribers, Twitter, YouTube subscribers, LinkedIn, and then Facebook. And then the most important is the messenger instant response you get when you message us. So WhatsApp, you can see Botang, Google Duo, Telegram, and then we have other platforms. Now we have a dedicated page on our website, where you can give on your information, your data, your details, whatever you provide on our website is secure, because we do not share the data with anyone. So and then we send the newsletters, and then you can get back to us, and with your queries, and all of these services are we are doing for free. So the most important thing is that we are a company which started in 2009, and then we have been doing online trainings and workshops since 2012. So we are not new to this. Like there are companies, there have been people who have started working online because of the COVID-19 pandemic, and then since March, or I should say May. However, we have been doing online trainings and workshops and research projects since 2012. So we have an ample of experience, and we have ample of ways in which we are used to this kind of platform. So previously, before the COVID-19 pandemic, we used to do 2 or 3 courses in 2 or 3 months, like free courses, what I'm talking about. But then, as this is also the 10th year, or I should say the 10th anniversary of BDG Life Sciences, hence in February, we thought that as it is a 10th anniversary of BDG Life Sciences, so why not to increase the frequency of free courses, and why not to launch free courses? So we started launching free courses in February as a gift of 10th anniversary of BDG Life Sciences. Then in March we got the lockdown, and then in since May we are all stuck up at home, and obviously attending the online sessions. However, I'm very happy to tell you or give you this information that since February 2020, we have conducted more than 75 courses, these free courses, and a big number of more than 14,000 participants have participated in these free courses from all over the world. So innovation and creativity is an integral part of BDG Life Sciences. That is the reason when we were used up with launching free courses and doing it again and again and again and again, then in the month of September we thought that why not to do something new, why not to do something extra. So the Envision with BDG Life Sciences initiative has come up, and then we decided that okay, fine, now we will gather speakers from all over the world who are experts and who are working into the industry, or who are good lecturers and professors from all over the world. Let's gather them, and let's arrange lectures, free lectures, so to give to the society, because we always believe in giving more than taking. So that is what how we are connected today, and you guys are over here, and you are getting this uh, free sessions. So that is how Envision with BDG initiative has come up. So I was telling you about the subscriber platforms. Then let's come to the business. What exactly we do, because business has to be the part of the company. So in 2010 we started the company Biodiscovery Group. We have, as we started with the group, so we have not removed a group from the name of the company, and that is why today when you could see BDG Life Sciences, then there is BD and there is G. So G stands for group. So this organization is for everyone. This organization, the aim of the organization is to help, and that is the motive of starting the organization. Hence BDG Life Sciences Biodiscovery Group is the organization which is for people. So what exactly we are doing? We are creating programs in such a way which leads to a win-win situation for both to us as well as the client. And this is what we have been doing. The research project program, which we started in 2010, like when we started the operations in 2010 March, then this was the first program which we started. And what we used to do, this the research project program, the we used to launch, in fact we are still launching novel research projects, and giving you an opportunity to work on a novel research project. So you pay us the fee, that is winning for us. And what you are getting training into the latest technology. Then you are getting an opportunity to work on a novel research project, which is also the practical application of the training, because without practical application you can achieve much greater heights. And then the most important thing is international publication by your name of that particular project, because as it is a novel project. Hence this is the win-win situation. Now the other thing is, it is opening a wide array of opportunities, like job opportunities as well as PhD opportunities for you. Because for example, if a person has joined with us in 2010, and that person was masters, but today that person has achieved greater heights of success into his future or into his career. For example, like he has become a scientist, or he has become a dean, and he is a research officer, and he wants someone for a PhD position. So he asked us, like, do you have someone? So we connect people who have recently joined with us. So we never advertise that our training program is a job assurance. You will get 100% job assurance. You will get, after doing this program, you will get jobs. No, it doesn't work that way. But I don't know why people advertise, in that way. Just after doing one training program, you will get a job. No, that's not possible. However, yes, what is possible? The most important thing is that when you work on strengthening your profile by getting international publications, when you work on updating your technical skills by learning new technologies, then yes, you have chances of getting much better opportunities in future. But if someone is saying that, I don't know why people advertise in that way, that, if you do this, then you get 100% job assurance. We never do that. But what we do is that we connect, because you when you join any of the program, then you become the part of the family, and when you become the part of the family, then definitely we connect with to you with other people. So this is what we have been doing. So what I was telling you about, that in 2010 we started our services, and our wide array of services include contract research, workshops, e-workshops, that is online workshops, research projects, online as well as on-site, and training, webinars, seminars, and conferences. So this is what we have been doing since 2010. Now you could see over here, this is what the business which we are getting. And because as it is a family, so probably you are new members of family, so you should know that how things, what is the revenue model of BDG Lifesciences. So we are getting a lot of people from the research projects program, because this is the best program which which is strengthening your profile. So that, and it is online, hence online as well as on site. However, these days everything is online, so this bar is the highest. Then we have conducted many workshops around the world, starting, if we start from west, then we've conducted workshop in Mexico, then moving again to Europe, Iran, and then Africa, Nigeria, Kenya, Sudan, Egypt, then coming to Gulf, Dubai, Abu Dhabi, then coming to Southeast Asia, where we have conducted workshops in Malaysia and Thailand. So, these are the workshops which we have conducted before the COVID-19 pandemic. However, there were workshops scheduled for 2020 in Amsterdam and Czech Republic, but then unfortunately we have postponed them. Then we have e-workshops. These are online workshops. Again, it has opened the window for participants from all over the world. So currently we have a molecular docking workshop that is going on, and then a structural bioinformatics workshop that is going on. So a number of workshops we are doing online. So you could just visit the website and see. And then these are the courses. So this bar has gone up just because of these free courses which we have launched as the 10th anniversary of BDG Lifesciences in February 2020. And then others, this is contract research, where we are doing a lot of contract research for research scholars and company professionals. So basically our mission is very simple. We want to provide solutions for the future by catalyzing the process of biomedical sciences into therapeutic technologies that benefit human health and society, and eventually fuel the economic growth. So this is our mission is very simple. Keep on working towards the betterment of the society. So these are the programs in which we are offering our services. However, we have included many more programs recently, like introduction to Python, and as there is a huge demand of database handling and data scientists basically, so that is why we have launched new programs targeting cancer research, in which we are doing programs like Python and R, and you could just visit the website. We've launched a number of programs. So this is what we have been doing, and you could see the cancer, HIV, diabetes, and tuberculosis research. We are focused on these 4 types of diseases in which we have launched our research projects, as well as workshops specific to these diseases like cancer targets, HIV, molecular medicine, HIV, then diabetes targets, and tuberculosis research. So moving forward, the most important slide of the presentation, and what I would like to tell you over here, that why you should come to BDG Life Sciences, why you should get training at BDG Life Sciences, what you will get over here which you will not get anywhere else. So first, I would like you to point out on these four words: innovation, ideas, service, and value. Innovation, ideas, service, and value. Innovation and creativity, I've already told you, is the integral part of BDG Life Sciences. That is why we keep on innovating and creating new programs and launching new programs. Then, if we will read it, then we will understand it in a much better way. Innovation is to translate ideas into service that creates value. So we are creating or translating our innovative ideas by giving excellent service to you so that it will be valuable for you. How? It will help you in your future. Now, what were the ways? Practical application of technology. Whatever training program or workshops we are doing, all of them are having practical applications. Then hands-on training sessions. We know that we learn by doing the things. Hence, we have started in 2010 the trend of hands-on training programs where our participants have to have a laptop or a computer system on which softwares are installed and they perform. So we have set up a trend. We are the trendsetter of hands-on workshops. And laptops, it's not about, you can install the softwares on your laptops and you can take back home. So what you are taking back home, you are taking back the learnings, you are taking back the, the softwares on which you can perform day and night, even after the workshop. And then excellent support system. This is the last point, the everlasting relationship. We will keep on helping you, we will keep on guiding you until unless you yourself don't want our help. So that is how I am calling this word again, family. We are a family and we keep on helping. Then novel research projects, giving an opportunity to work on novel research projects so that we can have international publications, and then result driven research so that we can publish it in good conferences, in good journals, and then everlasting relationship I already told you about. So this is the point, or I should say these are the points which outstand us with any other organization. So this is how we are different. This is how we are unique. So these are our achievements. You could see this graph is going up, and until unless this graph is going up, it does not have any problem, because once you start a business, and then if you do not innovate, then at one point of time it will come to a saturation point and then it will come down. But over here, as far as this is going up, this means that we are doing something innovative and creative. So in a nutshell, we have 10 years of 10 plus years of experience in training and workshops around the world, 60 plus research projects completed in which people like you have participated and got international publications, 400 plus hands-on workshops which we have conducted, 20 plus hands-on workshops in other countries I've already told you about. And then, we have conducted a lot of workshops, 50 plus, at premier institutes of India, like, for example, AIMS, that is All India Institute of Medical Sciences, New Delhi, IIT, NIT, VIT, MMC, JIPMER, etc. And then happy customers. Happy customers, the feedback. For a communication process, feedback is the most important part, because once if the feedback is not given, then the communication process is not completed. Hence, feedback is very important to us. Back then in 2010, when we started the organization, we used to get paper feedbacks, like we used to have forms and the participants used to fill the feedback form. Then we transformed it into directly taking feedback on our website. Now it is completely changed. Like, for example, if you attend our program, you need to submit a feedback video of yours. If you are not comfortable in submitting your video, then you can give your audio. However, we make that video and we put it on our YouTube channel so that people could see what is their experience, what they felt after doing our program. So that is a very unique way of getting the feedback, and we started and doing it successfully in 2000, since 2 or 3 years. And on our YouTube channel you could see a lot of feedback videos which we have posted. And before this session, today is 13th session, so all the previous sessions are already available on our YouTube channel. So if you want, you can go through them and see these lectures, as because these lectures, they are live on our YouTube channel. So as just quickly going through this, we were the first one to introduce online training in drug discovery and designing. We are also the first one to conduct training online and on-site in gene and genome editing using CRISPR-Cas9. We have conducted this training in May 2017. In 2016, this technology was discovered by Jennifer Doudna and Dr. Charpentier, for which they have, in 2020, they've got the Nobel Prize also. But, we were the first one to conduct. This shows the innovation and the creativity of the scientists who are working at BDG Lifesciences. So free courses, I already told you about. We've launched free courses to assist and facilitate the learning process during the hard times. And we've conducted more than 70 courses, free courses, in which more than 14,000 participants have participated from all over the world. So these are our services like virtual screening, molecular docking, simulations. This is for as a contract research which we are doing. Then NGS data analysis and genome editing, personalized training, and research project consultancy. These are very two unique programs. Personalized consultancy is something which is we want to help people who want to do training in specific topics. Like for example, if they want to do for their PhD, they want to do a training in certain topics, then they can tell us and we design the training program and we conduct the training. Research project consultancy is also for research scholars, especially the PhDs who are doing PhD. Like if they're stuck somewhere, then we can design the program and we can help or decide the future course of their project. So if you want, you can take the screenshot of this where we have mentioned our social media platforms, and you could help, or I should say, if you want any help, then the team is there who will be ready to help you. So thank you very much. And I will now stop sharing my screen. And Dr. Bharti, can you please unmute yourself so that, we'll ask you to, the team to make you the co-host, and then, you can carry forward from there.
Hello?
Yes, Dr. Bharti, we can hear you, but we can't see you.
Yeah. Yeah.
Okay, now you can start sharing your screen and please go ahead. So Dr. Bharti, I would like to, just introduce, as I have already said, that she will be taking session on classification of genetic variants and functional assessment. And she has a wide array of, experience and into genomics, and she is a computational biologist and provides leadership as a role. So she handles clinicians and patients and builds strong professional networks. She has a vast year, means 18 years of experience in, and she has gained expertise in molecular genetics. So, Dr. Bharti, thank you very much for this call. However, this session was scheduled before, and due to certain internet issues, we could not do it. But then now we are doing it. So, over to you. Please go ahead.
Yeah. Am I audible?
Yes, you are audible. Please go ahead.
Yeah. First of all, thank you, Dr. Asif, for a nice introduction. So today I am going to discuss about genetic variation and functional analysis. I have divided my topic into various subheadings. Introduction, which will cover variants, different type of variants, what are the different sources of these variants, and what is the significance of these variants, whether they are useful or not useful. Second section will include identification of variants, which will cover data generation techniques, file formats, different kind of tools which we can which will be using for identification of the variants and class into different types and the databases. The third section will cover functional validation, because once we identify the variants, it is important to validate in the biological system. So I will cover in vitro as well as in vivo systems. So let's get started. So what are the variants? So variation is the difference between the individuals within a species of a population. The variation can be the variation describes the diversity as a whole in the species or a population. The variation is important for adaptation and evolution, as we know that due to the changes in environment or the surroundings, some species will adapt to this environment. Why? Because of the change in the genetic makeup. So the genetic variation is an important force in evolution, as it allows natural selection to increase or decrease the frequency of alleles which is present already in the population. The genetic variation is advantageous in somehow to a population, because it enables some individuals to adapt to the environment, as I mentioned to before, while maintaining the survival of the population. These variations can be categorized into different types based on their characteristics. So first category is the continuous variation. The continuous variations within a population in which a graded series of intermediate phenotypes are present, as for example, height and weight. So as we know that the height ranges from some, say suppose 5 feet to 6 feet. So it can be 5.1, it could be 5.2. So it can range between the maximum and the minimum extremities. So these type of variations are called continuous variations. Another type is discontinuous variation. So it is not the between the extremes. It is only some kind of combination which is present, like blood group. So in blood group, only 4 types are available, like A, B, O, and AB. There is no other type of blood groups is present. So these kind of variations are called discontinuous variation. Another type of classification could be a somatic variation and the germinal variations. So the somatic variations, as we know, these are present in the somatic cells. So basically, these somatic variations are present mostly in the cancer tissues or tumor tissues, because of unlimited growth or several factors or the microenvironments leads to the somatic variations. Most of the time, these variations are de novo and doesn't pass to the next generation. Another type is the germinal variations. These variations are present in the uh, germinal cells like eggs and sperms, and these kind of variations, they pass on to the next generation. So they will keep passing from one generation to the another generation. So they are genetically inherited variations. Another category is based on environmental geographical variations. So as we know, some species or some organisms are restricted to one geographical locations. They are not present to another geographical location because of the environmental effect. So such kind of variations leads to the phenotypic, most of the time phenotypic variations. So now let us see what are the different sources of these genetic variations. So there are multiple sources of these genetic variations. So like gene duplications, mutations, leads to a new genetic variations which can be created within the population. A population, as for example, a population which is highly reproductive has a higher chances of getting the variation. As I mentioned that there are different sources of genetic variation. It could be mutation, which occurs when the sequences of DNA is changed. It can occur spontaneously by errors in DNA replication. They can be deleterious or beneficial. As for example, in agriculture, for high yield, better quality, resistance, or it can be a part of evolutionary process. The second type of variation could be arise due to the gene flow. The movement of genes from one population to another population is a cause through the gene flow. As for example, uh, it occurs mainly in the plants during the pollination. So if two type of populations are exists near to each other, and due to the pollination, like wind pollination or water pollination, so the pollen grains reach to the another population and cause the two sets of the genetic diversity or two gene pools in the two different populations. The third cause is the genetic drift. The genetic drift is frequency of different genotypes in a small population because of fluctuation of allele frequencies. These type can cause the disappearance of one type of alleles, that is a rare alleles, thus the evolution of new species. Another type is the sexual reproduction. As we know, during the crossing over or the recombination between the chromatids of the homologous chromosomes, it also can cause the variation in the genome. So there are different type of genetic variations. So in my previous slides, I have told you there are multiple sources of the genetic variations like mutation. So the mutation is a type of a genetic variation. So how these genetic variation leads to the changes and creates the variation. So there are large variations and the small variations. Large variations are because of the chromosomal changes like deletion, duplication, inversions, translocation. The translocation are between the two different chromosomes, or it can be a balanced translocation. So here in this presentation, we will be concentrating more on the gene related genetic variations, which could be due to deletion, insertions, or substitution, or copy number. Deletion means it can be one base can be deleted or another base can be deleted. Substitute one base can be changed to another base, purine to purine or pyrimidine to pyrimidine or purine to pyrimidine, or the copy number change. In this also the some sequences are inverse. So if the sequence of gene is changed, then also the genetic variation will arise. So now, sorry. So as I mentioned to you in my previous slides, to see these variations, how these variations are there in an individual. So which kind of, so how I am different from another individual, how a second person. So if I take a population of a human beings, how these each individual is different. So there are different approaches. So data is very important for us. So data generation, what is the process from where we will get the data to see the variation. So suppose if I would like to see how I am different from another individual, how one species is different from another species. So I need a data to see how they are. So there are two approaches to generate a data, either a Sanger sequencing data or NGS based data. So Sanger based, if I want to see only some genes or some genetic variants or just particular one variant, then Sanger will work. But if I would like to see the variation across the genome, then NGS will give us the better opportunity to look for the variation. So once we have the data, so when we have the Sanger sequences or the NGS data, so Sanger sequences, when we have the Sanger data, it will be in the FASTA format. So FASTA format is a sequences format, which is a very specific for Sanger. So we have to align them, we have to map to the reference or a specific gene, and different tools can be used like a bioedit, sequencer, UN gene, DNA star, mutation surveyor, you have codon aligner, we have multiple tools to do that. Here I am not concentrating more on Sanger variation analysis. I will be concentrating more on the NGS variation analysis. So when we have the NGS data, so first we have to see how our data is. So there are different tools to see that how our data looks like. So NGS usually generate the sequences in the form of FASTQ file. We will see later on what is the FASTQ format on what these files are. So just grossly, I will mention here the FastQC, FastP, NGS QC toolkit or CutAdapt can be used to refine or filter the reads, high quality reads, and for to get a refined data for downstream analysis or the variation analysis. So this is a typical workflow for NGS from the sample preparation to the analysis. Sample preparation step includes the extraction of DNA, quality check of the DNA, then the library preparation for different applications like WGS, WES, or transcriptome, whatever the application we use. The next step, once our DNA is ready, then library preparation. Library preparation step includes the fragmentation, adapter ligation, and barcode addition. Here, I would like to stress upon the barcode addition. This gives a very good, what you say, that a very good advantage over the Sanger, because in Sanger you cannot multiplex the samples, you cannot pool the samples, you have to process the individual samples. But here in library preparation for NGS, barcodes labeling will give uniqueness to each samples and you can pool. And after sequencing, you can demultiplex and identify each and every sample separately. So depending on your application, either if it is a WES or WGS or transcriptome. Transcriptome is a different application, so the library preparation is different. Here I will just stress upon WES or WGS. If it is a WGS application, no need of the enrichment, but if it is a whole exome, then the enrichment will be done by either a probe capture or PCR amplification, and the library will be amplified by PCR. Then again, there is a step for the quality check. The library QC will be done, sequencing will be done, and the raw FASTQ reads will be generated from the machine. So once we have these FASTQ reads, then the first step is to process for the quality check. As I mentioned you before, these are the different tools. And these are not only the tools, there are many, many more tools which can be used for the quality check of your sequences. The next step is the alignment. So if it is if the reference is available, then you have to just align your reads with the reference. If it is not, then the assembly. So different to do this job also, different assemblers are available. So if the reference is available, then BWA can be used, BOTI can be used. If the de novo alignment is there, so different pipelines, different set of tools are available. Then the variant calling. So now I will take it forward from the variant calling, because I assume that here everybody is not new and they know a little bit of the alignments and the references, what the terminologies I am just speaking about. So our main aim is to identify the variants from this. So how our sample, so there could be a two aims, how our sample is different from the reference genome, or what is the difference between the two samples. So that is our aim, to look for the variants. So these variants can be of SNP, single nucleotide polymorphism, or the single structure variants or the indels. Indels means here we are talking about the short deletions or insertions, not the large deletions or insertions. Then once we identify the variants from our sample data, then what these variations does. So we need to know, we need to annotate them. So the process is called annotation of the variant calls. So then once we know which kind of pathogenicity that they cause, then we can clinically interpret using the different databases. Then comes to the interpretation of the variants. So once we identify the variants, once we know what kind of pathogenicity they cause, whether they are pathogenic or they are benign, or they are likely causing a pathogenicity, or they influence other variations or genes to cause pathogenicity, in which pathway they involve, what other genes they are connected to, and what kind of, so all that process is called the interpretation of variants. So this also we will see what are the different tools and servers are available. So now there are different challenges also in calling the variants. It is not that easy, because once we have the alignment, as we know, we have exons, we have introns, we have UTRs, we have so many things in our genome. So when we will align and call for the variants, we will get some 50,000 or over 50,000 of SNPs, or some short deletions, some insertions in the samples. So they could be because of some base calling errors, maybe the technology what we use for the sequencing cycles, if the sequencing cycles are not appropriate. So all these steps needs to be validated before calling the variants, and we need to be sure that all these technology, what platform we are using. So our tools needs to go with the technological platform, sequencing cycles, and the sequence context. So what is our aim to, for what purpose we are calling the variants, for what purpose we are seeing the variants, whether it has some clinical significance, or we are doing for research, we are trying to discover some novel mutations, or so it depends on all these things. So low coverage sequencing, another step, very critical step, is the low coverage sequencing. So it is very, very important that if we are trying to look for some novel mutations, or some haplotypes, specific haplotypes, or some critical genes which are either located very closely to each other, or they have high repetitive regions, or if they have a high probability of getting the mutations. So we should have a high coverage of sequencing and even high depth of the sequencing. So because if the sequences are lagged from the two chromosome of a diploid individual, then we will miss the haplotype, true haplotypes, inaccurate mapping. This is also very critical step, so that our sample or our reads from the sample should be aligned with the reference appropriately. If the alignment is not good, if the mapping quality scores are not good, then it will either we will miss the mutation, or either we will report a miss mutations. So to overcome this problem, so once we have all these data generation guidelines appropriately, and we have sufficient data for variant calling purpose, so even there are good pipelines. You see, if we are doing for the clinical samples interpretation, not only for the clinical samples, even if we are doing for research, and so there is a very good pipeline available known as GATK pipeline. This pipeline is developed from the Broad Institute, US, and this pipeline can be used for variant calling from DNA sequences, RNA sequences, ChIP-seq also. And this tool has a very good, you say that very good script for variant calling and haplotype caller. So we will see in the subsequent slide what is haplotype caller and SNP caller. So SNP calling methods. So what is SNP calling methods? So first thing is that if we have to call for SNPs. So this particular pipeline is depending on the Bayes theorem. The Bayes theorem is based on the probability of an event based on the prior knowledge. of conditions that might be related to the event, current event. So this pipeline is totally on the knowledge base. So even if you have a low coverage data sets for the indels and all, this pipeline will accurately call for the variants. So haplotype caller, as you can see in these slides, there are multiple gaps in the original sample. So for when we align our sample reads to the reference, this is our reference. So when we align the reads to the reference, it will create some gaps and align, because the first alignment is depending on the global alignment. But so in GATK, what it does in the haplotype caller, so first the reads will be aligned on the reference reads using the global alignment. But after the correction using the haplotype caller algorithm, it will correct the alignment using the local alignment, and it repairs the gaps. Now you can see here in this this A. So if this is the incorrect haplotype, it calls here also. But when it corrects using the local alignment, it corrects and inserts a gap and then align properly and call for the haplotypes. Now this another step after that, once we call the once we call our variants, so we will get the VCF file in that. So we have to see whatever the VCF has generated, and it calls for the variants, whether those variants are true or not. So when we visualize something, we will get the confidence, yes, something is present or absent, just not by looking at the file. So when we visualize something, we will get confidence. So using these two tools, UCSC, IGV, IGB, we can visualize our files. So to visualize using these tools, we need a reference genome file, which should be in the FASTA sequence file, a reference annotation file, which is from the reference. So this we will see that what is the GTF, GFF, and that file it is, and the sequence alignment file, which is in the form of BAM file. So now we'll come to the file format. Let us look first what are the file formats we use and we talk about in when we call for the variants or deal with the NGS data. So first is the FASTQ format. So FASTQ format is nothing. So it is just the, so you have some 4 lines here for your FASTQ files. So here it starts with the A, at the rate of A. The first line starts with the at this symbol, and here you will have some name. So this line will contain your sequencer, your adapter sequencing, your index, and some unique name here. Then the second line will constitute your sequence. Third line will be your quality header, which will start with your plus sign always. And third will be your ASCII characters. ASCII character will define your quality of each base. So this format is called FASTQ format. While FASTA format starts with the symbol of greater than symbol, and it will be having your specific sequence name, that is called header. And the second line will have your sequences like a ATGC or something like that. So in FASTA file, this is in one line, it will be only 80 bases will contain, and then next line will start, whatever the length of your sequence is. SAM file is your sequence alignment file. So when we align our reads to the reference, so it reads, writes a file. So that is your sequence alignment file. So what it will do, it will write all the coordinates which read where it maps in the genome. So BAM file, then comes the BAM file. BAM file is a binary alignment file. It is nothing, it is just the binary form of sequence alignment file. So it compares, so each position is compared with the reference base, and if it is present, it will write 1. If it is absent, it will write a 0. So it's the binary alignment file. It's just 0, 1, 0, 1, and with the coordinates of with respect to the reference, and then writes in the file. So this is your alignment files after the alignment when you align your reads to the reference. So now the From this BAM file, we call the variants using our GTF file. Then we get the VCF file, that is our variant call file. So we will see in detail this VCF because this file is very, very important for the variant calling. So we will see this. So now how our VCF file looks like. A typical VCF file looks like this. So this will be a header and then it will have some columns. So usually, typically, it will have 10 columns, 9 to 10 columns. And then, so each column specifies a dedicated column with some specific notation. So the first column defines a chromosome. Second column defines a position. Third column defines a ID, RSID. Fourth column defines a reference base, whatever the base is present in the reference. Next column represents the alternate base. So what it will do actually? So it will not write each and everything in VCF file. So what it will do? It will compare your read with the reference. Say suppose your read is like this. Say suppose you have this. So you have, this is my reference. This is my read. So they will align like this, right? So what it will do? So VCF will take this read first because this is my first read. So it will compare this read to this region. Say suppose my reference has A. This position also has A. It will not write this. Now my reference has this base. Now this change is there. So it will write like this. So this is on the chromosome 1, second position of my reference, and this is my base at this position, and this is the base in my reads. So this is, it will write like this. Then we have to pass the quality filter. So everything. So if my base, this C is not good, how I can consider this is a variant, right? So what we will do? So we have to look for what is the quality of this base. Whatever is the variation is there, then we have to. So when we call for the variant, then that time there is an option in that tool that we have to set whether my all the bases should be above Q30 or more than 30. So that if the filter will pass, then only it will report. If my base quality is not up to 30, it will not report. So this is the quality filter, whether my base is a good quality or not. Then the next column is the information. We will see what these information means for. Then the format, then samples. So if you have the combined samples, 3-4 samples in one VCF, then it will write for each sample. So sample E1, sample 1, 2, 3, and so on. We will see for each, so these all this information and the format column, what it stands for. So now here you can see that. So now after my filter, my base quality is passed through the filter. So it will report some information. So if you can say that it reports for NS, DP, AF. So AF is my allele frequency. So here I have given all these names for this and what it stands for, it is mentioned over here. So you can go through that. And in the format also, it is the GT, GQ, DP, HQ. It means, so later on, if these are the quality features of your variant, what it stands for. So what is your read depth, what is your quality, what is your mapping quality of that particular base with respect to your reference. So all that does matter. So once we know all these things, again, it will help us to filter down or narrow down our variants from these files. So let us look what is the, so once we have this VCF file, how we can narrow down our filters, because in VCF file, it will report thousands and thousands of variants. which is not of our use or not of our concern. And every SNP cannot be a pathogenic variant or a abnormal variant, right? So we have to see using what is the frequency of that particular SNP in the community or in a specific population for which we are defining a variation. So this is the overall workflow from VCF file. Once we have the VCF file, so we have to filter out the common and low quality variants from this. So once we have filtered down, so we are narrowing down our variants from 50,000 to the more appropriate variants which may be likely to cause the disease or which could be linked to the disease where we are looking for. So then filter by inheritance or zygosity, right? So if say suppose we are saying that our disease is X-linked, right? So the variation should be present on the X chromosome. Or if we know that if any variation, chromosome 6 is linked to or 19 is linked to for some disease, so the variation should be present on 19 or 22 or whatever the chromosome. So like that, we have to filter out or keep them in the then reduce to non-synonymous or synonymous or missense, right? So we know that synonymous mutations will not cause any pathogenicity, right? So we can filter them out. So once we have the VCF file, we have to classify all the variants, whether they are non-synonymous, synonymous. We will see in subsequent slide how we can characterize them, whether they are non-synonymous or synonymous, which kind of a variation they are. And then whatever the remaining variants will remain after all this filtration, then we have to annotate and see what disease or what type of pathogenicity they link for. So how we can do this filtration of the variants? Say suppose in our VCF, when we filter out our low quality variants and other type of common variants, say suppose we have around 20,000 of variants. So now we have a database called DBSNP. DBSNP is a database where till date all the known SNPs are collected and kept in this database. So this database is linked for the population frequency in each type of population. What is the frequency of each SNP, whether it is linked with some disease or it's a common variant, whatever it is present in this DBSNP. So we have to, whatever the common variants are there, we have to remove using this DBSNP. Then we will be remaining approximately with 750 variants. So once we remove all these common variants and then we have to look for. Now there is another database called Genome AD or 1000 genome variants. This is also a database where low frequency variants can be kept and others can be removed. Then another comparison, that when we are comparing between two samples, so we will be having a control sample and we will be having a sample where we would like to see the exact variation which could be linked to a particular disease or whatever it is. So then we need to see which particular variants are present in both the samples. If those variants are present in the control sample, it means that they are not linked with any disease, right? So then we have to remove those variants when we compared with the control. So after this, we are again becoming lesser from these 20,000. So we are just having a 250 variants. Now these 250 variants are of our choice. So now we have to characterize these 250 variants, right? So this is the workflow and the process how we have to filter out our variant, and we have to keep only the variants which could be linked with some characteristics or some disease or some specific phenotype. So here we have to. So now we have only these 250 variants, and then we have to characterize whether they are missense, they are nonsense, they are splice variants, or they could be a synonymous. So as we know, synonymous won't cause any disease, so they also we can just keep them aside. So now out of these also, our major concern is for these three particular variants. So we will see one by one how we can characterize them and do the further downstream analysis. So now, how we can categorize our variants? Now there are two types of variant classification. So first is the variant classification, then the annotation. So what is this classification? So we have to classify our variants whether they are pathogenic or non-pathogenic, whether they are missense, nonsense, frame shift, or splice site variants. We have to see all these population frequency. What is the population frequency of these all these pathogenic, non-pathogenic, or so if say suppose I say that it's a pathogenic variant, right? But the population frequency is not mentioned anywhere. So this could be a novel pathogenic mutation in the sample. So if I say that, it could be my tools define as a variant as a pathogenic, but my frequency in some population is maybe more than 5%, right? So probably it could be a population specific variant. So we have to see all these things. So now once we identify whether our variant or the variant is pathogenic, non-pathogenic, which kind of a variant it is, then we have to do the functional analysis. So to classify our variant, there are different tools: variant effect predictor, DBSNP, polyfin. So we will see all these tools individually in subsequent slides, and functional to do the functional annotation for the variants. We can use David, Uniprot, KEGG, string, and cytoscape. So to do this classification of the variants, I don't know how you can go to this, but probably show the taskbar. I can take you to this. So probably I can put this link in the chat box, and people can go through this. And I will show you how so this is the link for variant effect predictor. So this is the tool. Here we can launch. It is a web browser as well. So when so it will give you this. So here I will start with the new job. Here a direct VCF file can be uploaded. So upload a VCF file. So I have downloaded, I have prepared one VCF file, and just choose, and there are multiple options over here. So if it is from mRNA, you can choose the transcript or Fseq transcripts, but as it is from the DNA exome sample. So I am just leaving all these as a default options. Then it will run, and it will classify your variants, whether they are pathogenic, which kind of a variant it is. So I have already done. So it will take some couple of minutes to do this job. So I can open this. So you can see that now this tool has categorized your variants. So how many variants were there? 16 counts were there. So it didn't filter out anything. So it means that your variants are good quality. So novel or existing. So 13 it predicts out of these 16 it predicted 13 are novel, 3 are already existing variants, overlap genes, transcripts. So it categorizes. It gives you visualization also, and then you can see over here it categorized all. You can download this file as a VCF file also, and as a text file as well. So this is our first tool by which we can classify our variant, whether it is a pathogenic variant, likely pathogenic variant, or VUS, or likely benign or benign variant. So this basically, if we are going for a clinical interpretation, this classification is based on ACMG guidelines. So ACMG guidelines are developed by the CAP, College of American Pathologists, and everybody has to follow while reporting on the clinical front. So the structure, function, and comparison with other species. This is important. So because once the variants are there, they are the part of the structure of the genome and structure of your the gene. And the initial phase of genome structure is done using the construction of map for the genetic physical map, and by which the gene identification, annotation of the features, and comparison of the genome structure could be done. The characterization of physical nature of whole genome, 3D structure of proteins which are encoded by these genes. So whether a protein coded by the normal genome, or what is the difference between a normal protein which is coded by a normal or reference genome, or coded by a disease genome. So that comparison is done by the DNA sequences basically, and then we have to correlate with the functions. What is the functions of these phenotype and the genotype? So this gives us the functional relationships, and then later on, once we have these structure and the function of a particular variant or a gene which is modified, or whether it's a part of protein structure domains or functional domains, then we can easily compare with the other species of the similar group or the order or family. So now let us look at the different software and tools. So there are various tools for processing the raw data to get the high quality sequences for the downstream analysis. So we will see one by one what are the tools which by which we can predict which kind of our variants are. What are the differences between different tools? So we can use a different combination of the tools or different sets of tools. So almost some tools are just complementary to each other. So let us see one by one what these tools are. So the first tool in this pipeline is the SIFT. SIFT is the sorting intolerant from the tolerant variants. So whether that particular substitution or base change, whether it can be tolerated or it cannot be tolerated. So the SIFT predicts whether an amino acid substitution affects the protein function or not. The prediction is based on the degree of conservation of an amino acid residue in sequence alignment derived from closely related sequences. So in this, what we do, we usually align our sequences, nucleotide sequences, translate, and then convert it into protein, and then using the sequences or using the SciBlast, align with multiple sequence alignment and download that multiple sequence alignment, and then use this SIFT, and it predicts how conserved is your amino acid change or substitution in your multiple sequence alignment. So it runs on this particular tool runs on the web server. It classifies the amino acid substitution, SNPs or indels. It can be interactively or via batch upload. You can download this tool and use your Linux or standalone also. You can you can upload your own single variant, or you can prepare a text file and upload directly. In this particular tool, you can upload your VCF also, and it annotates the variants which are present in the file and downloads the upload. You can download this as a text file. So I'll just go through this particular so we can see this particular. So we can go to this tool. I can explain you what this tool does, how it runs. So this is our SIFT tool, and here you can see that SIFT for genomes online submission. So non-synonymous. So there are different things. So you can just choose anything, whatever you would like to do. So this is how this is the database. And if you would like to annotate, so this so if you have your variant file, you have to choose your genome. So basically it runs for your Homo sapiens, and if you will see that whatever your sample format, file format, how it looks. So this is the each tool will have a different kind of a file format which it takes and defines you. So if I'll just copy this particular file and just paste it over here. And here you can choose whatever you want. You want ensemble gene ID, you want gene name. So once your process is, yeah, I will share all the links to you. So this is the status page. You can download your results from here. So it will download as a TSV file, which you can open anytime, anywhere. So it's a tab separated file, and you can open in Notepad also. So this how your files will be. So it will give you transcript ID, ensemble ID, everything it will give. So when you have this kind of a it creates a non-synonymous. So like that, it will define all your variants, which kind of it is. It will give you scores also, prediction scores. So when you want to compare with the different kind of databases, so these scores will allow you to compare between different tools. So which one have to consider, which one not to consider, all that will tell you. Okay. So I'll just put these things in the chat box also. I'll share. I don't know how to put in the chat box. I'll give you all these links in the chat box maybe after this presentation. So then you people can practice. I will share this file also with you so that you people can practice with these files later on. So as I mentioned to you, this you can this is your file format. You can upload a batch file also. You can upload 10,000, 2,000, many more variants together, and you can upload your VCF file also. Just copy and paste in a text file and then upload over here. And then you can choose whatever the information you need for your variants. You can choose it from here and then submit. And then it will give you a result output like this. And then it will give you a table like this where you can identify which kind of consequences your variants have. So another tool is proven, that is a protein variation effect analyzer. So it will tell you how your protein will affect, how it will do. So if so this particular tool, sorry. So if we go here, so before using each and every tool, see basically all tools will work. Okay, they will annotate, or almost every tool will give you all the kind of information. It depends on our aims and objectives, and we should know which kind of output it gives, which kind of input it requires. So I'm just taking you all these upload example, whatever the examples are there. So in this, you have to give the ensemble IDs. So ENS means ensemble IDs and your variation, whatever the variation is there in your gene, and then submit. So then it will give you once it is done. So either you can download or you can view your result table. So this is the kind of a table it will give. This is your input. It will give you what inputs you have given, what is the protein ID, what is the position, what is the residue in your reference, what is the residue in alteration in your sample. This is the score it will give. It will predict which kind of a variation it is, whether it is a neutral or deleterious. It will give you all that, right? So this is how you can analyze using that different tools. So the another tool is polyphen. It predicts whether your amino acid substitution or nucleic acid. See, this is all nucleotide. So there are tools. First, you have to convert your nucleotide DNA sequences to RefSeq or the amino acid. So it will give you automatically. It converts when you are giving your nucleotide variation. It automatically converts and tell you whether your protein will have amino acid substitution or not, and whether it is that change in your protein will be deleterious or not. So the next tool is polyphen 2. As we know that it is a polymorphism phenotyping version 2. So this is the link which I have given. It is a tool which predicts possible impact of an amino acid substitution on the structure and function of a human protein using the straightforward physical and comparative considerations. So it will compare whether your nucleic acid change is a purine or pyrimidine, or your because of that your amino acid, which kind of amino acid is changing, whether it is acetic amino acid to basic or basic to organic amino acids, which kind of amino acid is changed. So because of that, what kind of a changes it happens. So it predicts based on those properties. It also runs on a web server, and you can download this particular tool and runs on your computer also. It classifies amino acid substitution or SNPs. It requires again a simple text file describing your variants. So this is a kind of file format is required. This is the kind of output it will give. Again, it will give you the position, which kind, whether reference based, alteration based, and then it will predict your changes, whether it is a probable damaging or which kind of variant it is. So again, I will tell you these different kind of tools will use different kind of terminologies. Some tools will use pathogenic, non-pathogenic, benign, or probable benign. Okay. Some tools will use the same thing whether it is a damaging or non-damaging. So we have to be careful while using these terminologies, which kind of terminologies each tool use and how to correlate them together. In this pipeline, the next is mutation assessor. And this is the link where this mutation assessor is available. Again, it also runs on the web server. All these tools are freely available, open source, and well validated. They have a number of publications, good labs, good academic institutes. They're using this and putting in their publications. So all these tools are open source tools and well validated. So don't hesitate to use them. But one advice is that how to use it and where to use. It all depends on our aims and objectives. So mutation assessor also predicts the functional impact of amino acid substitution in the proteins. The functional impact assessment is based on the evolutionary conservation of the affected amino acid in homologous proteins. It is designed specifically for evaluating the somatic variants in the cancer or tumor samples. The variants are referred as functional rather than damaging or disease causing. As I mentioned to you before that different tools will give you different kind of terminologies. So this tool will tell you whether it has any functional impact or non-functional impact. While before, whatever the tools I have mentioned, they will describe as a damaging or non-damaging or disease causing, pathogenic or non-pathogenic, right? So these terminologies are complementary to each other. They are similar, more or less similar to each other, but not very different, not very similar. So concentrate on these, how the output is, how we are reading the outputs. So this mutation assessor captures the variants with the various consequences. It captures for the loss of function, gain of function, drug resistance, switch of the function. So these tools will give you whether it will list whether this for that particular variant linked to kind of a disease and if the drug is available or not, whether that particular variant is having a drug resistance or which kind of a function it will, whether it will switch for the function of the gene or not, or it impacts on other genes. So those kind of information this particular tool will provide us. So how usually it works, it will again work as a multiple sequence alignment. It calculates the score for each amino acid substitution, and it will calculate the conservation score and the specific. How specific is that particular amino acid for that particular gene or the species, and how conserved across the families. So it will adds both these scores and then works and then provides a score and classify whether it's a functional or non-functional variant. So this is how it works. So you put your variant over here. So again, when you will look at the file format, here you need to give the gene name and then the variation. And then you submit the file. Again, it can take your VCF file or a text file, and then it classify your variants like this. So whether you have whatever the functional score is there, right? All these it classifies on based on your conservation score of that amino acid and the uh, specificity of the amino acid in the protein. So in the pipeline, the next tool is mutation tester. This is the link where it can be assessed, this particular tool, and it integrates the information from different biomedical databases and use established analysis tools like polyfill. So backend, some tools, maybe the name are different, but at backend, it takes the information from the existing or existing databases or existing tools, other tools. So this particular tool also fetch the information from polyfill. So the analysis comprises the evolutionary conservation, splice sites changes, loss of functional protein features, and change the effects of the amount of mRNA. And it also predicts the disease potential using the naive Bayes classifier. As I mentioned before, this is the trained algorithm which is based on the knowledge of the prior existing variant. So this is as I mentioned that this is a trend classifier based on the 50,000, over 50,000 disease mutations and 520K common polymorphisms are gathered in this database from the various sources. And then it gives you it classifies your variant, whether it is functional or non-functional. It is basically based on the DNA alteration rather than the amino acid substitution. It annotates indels also and the non-coding regions in addition to the exons or coding SNPs. It combines adjacent mutation into a complex substitution polymorphism of to determine the true amino acid change. Say suppose if we are just aligning from the middle or maybe just the exons and some part. So we know that the splice sites has a plus minus two bases, upstream and downstream of any exons. So if the amino acids are coding from there, then it classify that. So it consider the substitution of the nearby bases of the exons or the amino acid changes. And again, it runs on the web server, and directly your VCF file can be uploaded to this tool. No need to prepare any extra files for this particular tool. So this is how the tool will. So you can upload directly your file, and then it classify your variants, whatever is present in your VCF file. So reference, alteration, whether it also provides you whether the particular variants which you are looking, whether already existing in the CleanVar. So CleanVar is a database where all clinical variants are present, which are linked to different or various diseases. So it reports along with that other information into this. So this tool is very good if somebody is working on clinical data. So it will fetch, it will classify your variants, and it will provide you an overview whether that particular SNP or DEL or type of splice variant already existing in CleanVar and linked to some disease. So the next tool is FAM. So this is enhanced accuracy in predicting the functional consequences of non-coding and coding SNVs. So this particular tool classify the functionality through the hidden Markov model, that is HMM, and it is again a web server tool. So it predicts the functional, molecular, and phenotypic consequences of the protein missense variant. So say suppose if you can filter out your only missense variants and use this particular tool to classify further your variants for the functionality. It utilizes the alignments of homologous sequences and conserved protein domains as a backend. So this is the mutation tester. You can put your variants here, and so because this is only for the missense, and it classifies your pathogenicity of your variants. So it gives you in a different color so that you can visualize immediately that whether it's a pathogenic or non-pathogenic. So pathogenic will be highlighted in red, benign predictions will be in green. So it will give only two types, whether it's a pathogenic or benign, whether it will link to some pathogenicity or not. So it won't classify in between. But looking at the score or combining different approach also, we can consider that whether it is pathogenic or could be linked to some disease or not. So the another tool is the SNP-EFF. This particular tool performs the both the functions, or this tool can classify your variant effect and as well as annotate also. So in this, there are two algorithms, SNP-EFF and SNP-SIFT. So SNP-EFF classify the genetic variation uh, and annotates and predicts the effect of genetic variants of the genes and proteins. So once we filter out our SNP, so say suppose we are filtering our SNP. So using SNP-EFF, we can filter SNPs and indels. So we can set our depth of the reads there, how much read depth we want to keep our variants or not, or indels. Then once we have filter out our SNPs and indels, we can use SNP-SIFT to annotate the genomic variants using database filters, manipulates. So this is all working at the back end of this SNP-SIFT and annotates the variants. So one annotation is done using SNP-EFF. SNP-SIFT helps to filter the large genomic data sets. So this tool, particular tool, is a standalone tool you can download, and basically it runs on Linux platform. But it is easy to use and it is well validated. And in one toolbox, you can filter also, and you can annotate your variants. So this particular tools classify as a pathogenic, non-pathogenic, synonymous, non-synonymous together. So next is the splice finder. So this is the tool which you can use. So in this, say suppose when you are aligning and you find that, okay, this particular sequence or reads you have the variation or variant, you would like to know whether that particular variation in the splice sites or not. So you can put that direct sequence in this tool, and it classifies whether that particular variation is involved in these splice sites or not. Because if we know that if alternative is like if these splice sites are shifting, so the structure of the protein and the function of protein will alter. So the next is the pathway network analysis. So to do that, once we identify our variants, we know the genes using different tools, we know the effect of them. So now in which pathways they are involved, what all pathways will get affected. So there are different tools to do pathway analysis like David, Uniprot, KEG, String. So all that you can use for the network analysis. So once we identify pathways, but in one pathway, there are multiple genes, multiple genes, they interact together. So we should know that how these genes are interacting together. What is the network of these genes? How, whether only one gene is getting affected, one protein is getting affected, or if they are interacting with different genes, different proteins are getting affected. So that is a type of analysis is called network analysis. So this also can be done using cytoscape or String. And again, once we know all these variants, how they are, once we validate, yes, this particular uh, variation is present in different tissues, in different samples, or in different sequences. So we can visualize again using either Cytoscape, UCSC Genome Browser, or IGN. So now let us see the different databases. Okay, now we have identified our variants. Now we know this particular variant is whether it's a splice variant, or it is a pathogenic, or it is a synonymous or non-synonymous missense variation. Now we need to know what is the evidence of that, whether already those evidences are present or not, or it is entirely a new variant which is not even yet reported. So we should know that. So then only it will be easy for us to correlate with the disease. So suppose if we are looking for some lung carcinoma, we would like to know that whether P53 mutations are all which are already reported as a germline, whether they are there or not, which could be the pathogenicity in that. So there are different databases where different kind of variations are already reported. So in that line, first is the genome AD. The genome AD is a database. It's a largest database, and it is built on the exact. So earlier the database was exact. Exact is a exon database. So whatever the variations are present or known till date, all the variations are reported in exact with link to the disease, different kind of disease. Now exact is not working, so it is shifted to genome AD. So all these genome AD, whatever the genes are there, you just put your gene on, you can see the variation, you can filter the variations, and you can see. So another is the human gene mutation database. Here also you can see all these mutations which are present or linked to the disease, phenotypic variations. Another is the lead and open variation databases. So within this database, you have some databases for specific disease. You can filter, you can search by genes, you can search with your variants. So CleanVar is specifically for the clinical samples. If you would like to see your variants is already known to some and linked to some disease, so you can use this particular database. Somatic cancer databases are there. So then variant interpretation for cancer consortium. So these two databases are having the phenotype information also. So genome connect, basically it is having a patient portal. So if any patient would like to see which kind of variations they have, which kind of treatment they can get, where they can get, what kind of other information they can get, whether already known patients are there, so they can just upload their report, and they can see everything. They will get a support from this particular database. So this is a list of the database specifically for the cancer tissue sample. So this is all. So if I can say that gene drug knowledge. So if you have a particular variant related to the cancer, whether the drug is available or not, which kind of a drug is available or not, whether known patient sample is there or not with the same mutation, which kind of drug will work, which kind of drug will not work. So all that information is available in different database. Just it is the matter like we should know that what all databases are available, and which database we will get the information of our interest. So before doing all these analysis, structural and functional analysis, we should ask questions to ourselves. What questions we are going to ask? First thing is that what kind of variants we have in our sample, whether those variants are already reported or not. If reported, which kind of functional abnormalities they cause. If not, which it could be. What kind of abnormality or pathogenicity it can cause. Whether so, if we once we know the uh, pathogenicity or variation they are, then we can ask that whether drugs are available or not, which kind of drugs will work with these particular targets. So these kind of things will give us this space to discover a new targets for the drugs, or discover a new drug molecules for the same targets for which already drug is known, because sometimes one particular drug will work for one individual, not for another individual. So this kind of analysis could be very precise and will work for the precision medicine. So now the another things. So now we have looked at different bioinformatics tools to predict our variations. We know which kind of a variation it is. So this is all bioinformatics or in silico tools to get the information which kind of variant it is. But it is very, very important that it should be biologically validated. Right. So then only we will know that, okay, yes, this is the function of this variant. It caused the pathogenicity. So biological functional validation is very important. So we have several current challenges in doing that to assess the functional impact of this set of mutations. So if it is a single mutation, it can be very easily correlated. But say suppose if you have a set of mutations, 2 mutations together, 3 mutations together, how a biological impact would be. So this is a current challenge in functional validation. The another one is that the complement the identification of functional and driver mutations by the prediction of how mutations affect protein and cellular function, right? So say suppose if the driver mutation is there, we predict that with using different tools. So we could come up, okay, this is a driver mutation. But how we will get to know that what which gene it is causing, and it is affecting the other genes and the cellular functions of because of that variation. And application to predict the tools of biologically relevant questions such as drug resistance, right? So how drugs will get resistance for that particular variations. So these are the current functional challenges are existing. So to do that, we have different type of models uh, for the functional validation. So to do that, we can do that, as I mentioned in my previous slide, that there are two kind of systems to do the functional validation of any variation, in vitro and in vivo. So in vitro, in vitro is like a 2D or 3D cell line culture. So say suppose if we would like to see the effect of any variant in, so we should have a prior knowledge of that. If our variant is in the, say suppose epithelial cells or lung cells or prostate cells or specific to any tissue, then we need to create that particular variation in the cell line, so then we can see the effect. But this is not a very accurate, because this is a very very systematic system, very organized, because we are feeding the nutrients, we are controlling the environment, so the effect which would be predicted is less accurate as compared to the biological system. So to overcome this problem, we have different kind of biological system like bacteria, C. elegans, Drosophila, zebrafish, mouse. So these are the human disease modeling models these days, where we can generate the pathogenicity as close to as a human, and we can create the mutations inside the system using different technology, using either a CRISPR-Cas or mRNA or miRNA, siRNA. We can create the mutation inside the system, and then see the effect of drugs, whether we will be able to reverse these pathogenicity or not. So this is the comparison will give you overview how a mouse system is different from the cell culture and the zebrafish, Drosophila, and C. elegans. So each system has their own advantage and disadvantage. So if I consider that mammalian cell line culture, so again it's 3 to 4 weeks culture, mammalian cell line culture, and but it is a very again systematic one and very organized and controlled environment, where it will give you an overview, or it can give you when you test the drugs whether it will work or not. But how and till what extent it will work, we do not know, because it can fail in the biological system, because biological system, different protein, different genes, they work differently. So mouse model comes. Mouse model approximately 3 months is life for this mouse, and they are more close to human beings. But the disadvantage is that to rear these, to create the mutations, again, it's a difficult task and the maintenance is very difficult and costly. So to overcome these problems, now there are multiple, because the even the fertilization rate is also very less in mouse. So then zebrafish, Drosophila, C. elegans are these animal models which are gaining importance to do the functional studies. So first comes the C. elegans. So the advantage of the using the C. elegans for human functional modeling, because if they are rapid and precise gene manipulation, they can replicate in a much faster way. It can be reared in the lab conditions. They can be used for phenotypic assessment and the cell-specific manipulation to determine the origin of pathology can be done. Gene function studies can be done. It can create the molecular networks, insight into disease pathology, and they are also could give a very good insight in understanding the clinical value, because they whatever the phenotypes we see for related to a particular disease in the human beings, they also present that. So they are good model in understanding the clinical variability, and they so these targets can be used for a therapeutic purpose for the drug development. So the mouse model basically used for cancer gene validations, cancer gene, host pathogen interaction, cell of origin, biomarkers, because these mouse is very closely related to human Homo sapiens, so they are still widely used. So zebrafish, again another upcoming animal model is zebrafish. So we can model the human disease. Basically, again, it's a cancer or leukemia. It's very becoming very common and popular. It share most of the organs with mammalian counterparts, and differently aged animals offers distinct advantage for cancer related pointers. So again, whatever the phenotypes we see in cancer, they also could replicate the same kind of phenotypes. So it is very closely related. As for example, in DBA, DBA is type of a cancer. It's a bone marrow, diamond black man anemia. So in this, if this anemia is there, we can see this particular phenotype in the zebrafish also. So here, as you can see, this is the anemic. So when we give L-leucine and it reverses the phenotype, so again it retains. So it recover from the anemia. So this is a very good example of human disease modeling for DBA. So I would like to just inform you here that there is a consortium of rare genetic and bone marrow disorder, which is functional now, and trying to create a network for this repository, because so we have a very good facility at this consortium, and this is the link for the registration. Any student or any professor or any academician or clinician is interested to be a member, so registration is free of cost. They can register. So this particular fish model is a freshwater fish native to southeastern Himalayan stream. They are rapid embryonic development, about 3 to 4 days only. They are large, robust, and transparent embryos. So embryos, usually fish embryos, are small, but zebrafish has a very large embryos. So you can see actually what is happening in that. So visually, if you would like to visualization the change, you can very easily see in that. And these embryos develop outside the mother, not inside the mother. So this demonstrates similarity to mammalian models specific to human in toxicity testing. So these models are now using in the in drug development. So we create a mutation in the zebrafish and test for the various drugs. And the main advantage of that, the drugs can be added directly to the tank water, because the water tank where they rear. So we can add the drugs directly into that tank, and with the water they can take it up. And another advantage is that zebrafish, the reference genome, the fully annotated sequence reference genome is available. And now there are multiple mutants and transgenic fish lines are available, like a cell lines. So we have a repository for cell lines in ATGC or ATCC, and another repositories are there. So there are repositories in various places where these mutants and transgenic fish lines are easily available. So like DMD is there. Another many genetic disorders are there where you find the missense. You can create very easily the mutations in zebrafish and then use for drug development or understanding the pathology of the disease. You can do many more things using this model. So I insist for the students for study purpose. This consortium also provides hands-on. If somebody would like to learn how to develop a zebrafish model, how to work on zebrafish, so we provide training also, and we have a very good training program. And if somebody would like to do the PhD on zebrafish, how to develop these models, mutants, they can come and learn. So this is the link. Anyone can go through this and see what is available over there. Another good model is a fruit fly, that is a Drosophila melanogaster. So this is also like it replicates. So where the organism replicates in a much faster rate and the life span is very less, and we can generate the mutants very easily. We can generate 2-3 mutants in one organism and use it. We can see the interaction of these mutants, particularly how they interact together, what kind of intensity they produce, what kind of pathogenicity they produce. And these are the organisms where we can test the drugs also, multiple drugs also. Because when you even create the cell lines or high throughput sequencing, high throughput chemical screening, because they are happening in the controlled environment. So this will be again, when it comes to the biological system, it will be different. So that's why these models are coming, because they are more closely to the humans. So they are more popular, becoming more and more popular day by day. So there are different variant calling applications in rare disease. So if we know that, okay, we are looking for some rare disease which is not very common, so, and we call for a variant and which could be linked to this particular disease. So if that, so it could be either way. So common variant which is having a very less frequency in some population or some in rare diseases, or it can be a rare variant which is common in some population, or vice versa. It could be the exome sequencing. So these are the kind of variants which are present in across all the coding genes, which could be linked to definitely to some proteins or diseases. Then the whole genome sequencing, mitochondrial diseases. So even a single base change in the mitochondrial genome will lead to some diseases. So that is their variant calling is very, very important, because mitochondria is just approximately 16.5 kb genome, and each base has its own importance, significance, and relate to the disease, different kind of diseases. Another very good application of variant calling is quasi-species identification. So as for example, in RNA viruses, so these viruses, RNA viruses are mutated, gets mutated very easily. So say suppose HCV, HIV, these are RNA viruses of clinical importance. So when a person or a human being gets infected because of these viruses, RNA viruses, and the patient is on drugs, so this virus itself get mutated and try to gets the resistance towards this drug. So why it is happening? So it is not happening because of the changes in the genome or some subspecies or a strain. It gets mutated and try to get resistance towards this particular drugs. So mapping the variation or mapping the mutation spectrum in a patient samples, how over the period of time a genome is getting mutated at different genes and different positions. So if we map that then we will get to know how we can overcome the problem of the drug resistance. So quasi-specific, because you will have a viral load and other things. If the viral load is very low, then it is very difficult to call the variants in different at different time points. So deep sequencing is really important, and you need to generate a lot of data for this quasi-specific identification. Another very good application is the transcriptome analysis. So if someone finds variation or or a kind of SNP or indel directly in the mRNA sample, so directly we can go back to the DNA sequence also, and we can go forward also for the protein prediction, how the protein structure and function will be modulated because of these variations. And then it will help us in predicting the drug outcome. So with this, I will just stress on the take-home message. The take-home message would be there are plenty of prediction tools are available, as I mentioned to you, and many of them will have a common feature. And most of the tools, they are rely on the alignment, multiple sequence alignment. This is the key for all the tools, maximum tools. So understanding the output and reliability, we would be able to categorize the variants more appropriately. And it is very, very important to have the complementary approaches which we should use to define the variants. So with this, I would like to end my talk over here, and we can take the question and answer.
Thank you, Dr. Bharti. It was a really nice and amazing session. So, thank you for your efforts. You have taken out time from your busy schedule, and it's really very nice to see that you have explained all these things which are used in NGS in such a easy manner. So, I hope and I feel that everyone will definitely have learned something, and thank you very much. Now I don't see any questions over here, so if you have any questions, then you can put it in the chat box, or else we will just wind it up and finish up the session with sending you the feedback form link, so you can fill the feedback. And this feedback form will be active only till 1 p.m. tomorrow, India time. So you can fill the form, and we will be sending your certificates probably day after tomorrow. So thank you very much. And people are already sending their wishes to you, Dr. Bharti. Nice presentation. So thank you very much. And let's see if we have any.
Even probably Dr. Asif, you can share my mail ID or whatever it is. Then probably if someone has any questions or
Important,
would like to learn more, they can approach.
or the most important thing is that the links which you have sent, what we will be doing is that you send us the links, okay, and we will post it into the
Yeah.
WhatsApp group which we have created for this particular session. So anyone who is interested, people, they leave WhatsApp group very fast. I don't know what exactly the problem is, but then it's okay, no issues. But you send us the links. Okay, you just please, Dr. Bharti, you just take a note and send us the links, and we will post it
Yeah.
in the WhatsApp group, so that the people who have attended the session, they
Yeah.
can access it. Okay.
Yeah. Sure. Sure.
Sure. So thank you very much, Dr. Bharti. I think there are no more questions. One question we are getting from YouTube: what softwares do you prefer for alignment?
See, it depends on your platform, which platform, sequencing platform you use, and the read length. So basically, if you have 150 to 200 read length, so then you can use BWA. BWA MEM is very good aligner for short reads. So you can use Bowtie also. And for RNA, there is a different aligner. So you can use this also.
Yeah,
But see, every time you go and search, there are new alignments are coming up. So based on your data, you have to validate, you have to search and then validate and use it. But
Yes.
most commonly BWA is used.
In the whole genome sequencing and the RNA-seq as well as the DNA-seq, we are using, in RNA-seq, we are using, I think, top hat and then bow tie and
Yeah, top hat is for it's a complete pipeline. It is just the because for RNA you need to use the splice aware
yes,
aligner. So top hat is very
mixing
good for the
junction.
reference base. If it is a de novo, then you can use Trinity. So it's entire pipeline you can run.
Yes, 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 yes. And for DNSing, we are using bow tie. So yeah, I think it's working fine. But then these days, it's have a Galaxy servers are having problems. So there are a lot of people who are using Galaxy servers.
You can use BWA. BWA is a very good. So if you are using exome data, BWA is a very good aligner.
Right? That is okay, fine. So thank you very much. That is one question which we have got. And that is my pleasure. So, Dr. Bharti, thank you very much. And thank you all the participants who have attended this session. The feedback link, as it has already been told, that it would be shared only in the for the people who will attend the session on Zoom. So it is been shared, and that is it. So thank you very much. Please provide your valuable feedback for the session, and thank you, Dr. Bharti, and thank you all the participants. We have only two more sessions in this Envision with BDG initiative, and one is Future of In Silico Drug Design by Dr. Hassan from South Korea, and one bioinformatics session. So just please visit the website and you can register for it. So thank you very much, participants, and thank you, Dr. Bharti, for your time. Thank you very much.
Thank you so much.